Stone Cellar Brew Pub across the river. So a lot of my work over there involves translational brewing. Now, what is translational brewing? Translational brewing is essentially the bridge between the laboratory and the brewery. So how do we take scientific principles and translate it so that industry can use it? And a lot of this has to do with the fact that we've been using yeast as a model organism for years and years and years in such fields as aging research or cancer research. And we can even get it to produce certain proteins for us, like insulin. And at the moment, brewers aren't using a lot of the knowledge that we know about yeast to brew beer. So, theoretically, we can use a lot of the information that we already know to figure out ways to improve our beer. But before we talk about what makes a better beer, let's talk about what is a good beer. So what is a good beer? The first thing that you notice when you open a beer, or before you even open a beer, it's going to be sight. So what type of packaging it's in. Here we have an example of 3 Floyds or Stone. How much foam and how stable the foam is. What type of color the beer is. And then finally we get to flavor. And my particular interest is in flavor because although all of the sight cues are really important for determining whether or not you pick up that beer, what ultimately makes you come back is the flavor, whether it's good or not, and specifically how consistent that flavor is. So let's take an example. Let's say that you open a beer and you drink it and it's wonderful. Great. A week later you're like, I really want that beer. You come back to it and it's not the same, chances are you're probably not going to pick that beer again. So, where does a lot of this flavor come from? Well, a lot of it comes from grains in the form of sugars that can be caramelized. It can also be processed to give certain attributes like chocolate or smoky. And then we also get a lot of flavor from hops, some bitterness and some aromatics from dry hopping. But a lot of it comes from yeast. And it's important to note that the yeast that we use for brewing has been bred for its flavor for potentially hundreds and hundreds of years. So over the course of the 1,000 or so years that we have been brewing beer, the specific strains of yeast have been selected for specific flavors. So a couple examples of some beer styles. We have a Belgian, and some words that you expect to see with Belgian is like fruity or spicy or malt forward. Or IPA, which is an India Pale Ale, which is very, very hop forward. German Pilsner, which is going to be crisp and bitter and clean. And one thing to note is that each one of these styles requires a different beer yeast strain. And we're not exactly sure what it is about these yeasts that impart some of the flavors to the beer. Although we may not know exactly how they're doing it, we do have some good indicators as to what molecules are actually causing certain smells. For instance, this is isoamyl acetate, and it tastes like banana, or it smells like banana. And in particular, it's this functional group here, the ester, that imparts a lot of fruity flavors into beer. A couple more would be acetaldehyde, which tastes like green apple, and diacetyl, which tastes like microwave popcorn. So if you put a bag of popcorn in the microwave and you let it go and you open it up, it's that smell. And actually, I have an example here for you guys. If you guys want to pass this around, and if you're brave, this is an example of diacetyl. I would not stick my nose directly in it. So, the next question then becomes, how does yeast create all of these flavor compounds? Well, it comes down to their metabolism. So acetaldehyde and diacetyl are both byproducts of yeast metabolism. So before we start to talk about exactly how these flavor compounds are being created, we have to kind of discuss some of the players that are involved in yeast metabolism. The first being glucose. This is an example of D-glucose. And it's just a monosaccharide. This is the basic unit of, it's the basic unit of, yeah, it's really strong. It's just the basic sugar that the cell turns into all of its energy molecules. The next one would be ADP and ATP. which is just basically the energy currency of the cell. CO2, which is just essentially a metabolism byproduct. And then finally, NAD plus or NADH, also known as vitamin B3, which is an electron carrier for this cell. And it also functions as its reduction power. And a lot of the work that we've done centers around this molecule in particular for this reason. So just an example of reduction. Reduction is going to be the addition of electrons, essentially. So in this case, this is the reduction of diacetyl. And we have diacetyl here. This is a carbonyl group, and it gets a pair of electrons and a pair of hydrogen atoms and becomes reduced, and then it is further reduced into 2,3-butanediol. So now that we've kind of discussed some of the players, this is pretty much the fate of glucose. Glucose is ingested into the cell and then is converted into pyruvate through the process of glycolysis. And in this process, the oxidized form of vitamin B3, NAD+, is converted into the reduced form, NADH. From pyruvate, there are a couple of different pathways that you can take in muscle cells. So for instance, for a lot of you runners, once you run out of oxygen, you start to ferment, you start to produce lactate. So if you get that burning feeling. With oxygen, you end up turning pyruvate into acetyl-CoA, and then ultimately into CO2 and water. But you also get some NADH out of that process as well. The exception to this is going to be in yeast when we convert pyruvate to ethanol. This is actually going to give us some of the oxidized form of vitamin B3. So it becomes, the fermentation process becomes a way for the yeast cells to take reduced B3 and turn it into oxidized B3. So what other roles does NADH, what does it do in the cell? Like I said, it's an electron carrier, and its primary objective is to create this proton gradient within the mitochondria. So essentially, NADH drops off its electrons into the electron transport system, which then creates a proton gradient, essentially pumping these hydrogen atoms out into the intermembrane space, which then can be used by an ATP synthase to create ATP. So essentially, it takes the electrons and it hands them off to the electron transport system to create a gradient and thus create energy. Well, what else can it be used for? Well, let's go back to this reduction of diacetyl. So we have two reduction reactions happening where we're creating two alcohol groups from two carbonyl groups. And if you add in NADH, suddenly you realize that it's also important in the reduction of this compound. Now this is important because diacetyl is by and large considered an off flavor in beer. However, after it gets reduced to 2,3-butanediol, it has no flavor, or it has no taste whatsoever. So it takes something that is very pungent and very unappealing in beer and converts it to something that we don't notice. So we've talked about NADH being important in energy creation. We talked about it being important in flavor compound reduction. Can it be implicated in anything else? And actually, what they found is that it can be implicated in aging, specifically using a system called calorie restriction. In this system, you basically reduce the amount of glucose that a yeast cell has, and it's been found that it increases longevity. Longevity being determined in a yeast cell by how many times it can replicate before it dies. So if you look at this guy over here, this is our lovely yeast cell, our mother cell, our daughter cell, and then you can see these budding scars, and that's how we tell how many times it has been replicated. So how does NADH fit into this? Well, it's been found that during calorie restriction, you have a higher ratio of NAD+ to NADH, which ultimately imparts part of this longevity from calorie restriction. Okay, that was a lot of science. So we're going to translate that over to the brewers. How do they see this NADH aging and reduction of flavor compounds? Well, the first thing is that they notice a tired yeast. The beer stays a little greener longer, so it has more diacetyl and acetaldehyde. It may under attenuate because it may be aged in it, or it may be tired yeast equals aged yeast, and it can be some of the side effects of that can be under attenuation, so the yeast are not eating all of the sugars that they could be, which is problematic for a couple of different reasons. But it also, and premature flocculation would be another indication of that. But it's also important for flavor compounds, such as diacetyl. So if you recall, if a yeast is aging prematurely, it's not going to be able to get rid of all of this diacetyl during its diacetyl rest. Okay, so how do we know what the NADH is being used for within the yeast? Well, one of the ways that we can determine this is by measuring mitochondrial membrane potential, or MMP. And mitochondrial membrane potential is essentially a measure of the electrochemical gradient in the intermembrane space. So if you recall, one of the roles of NADH within the cell is to pass off its electrons to the electron transport system to create that proton gradient to generate energy, right? So what exactly is potential? In this case, it is just a difference in the number of protons. It takes energy to take protons from the matrix inside the mitochondria and pump them into the intermembrane space. An example, I have this ball. If I hold it above my head, it takes energy to hold it above my head, right? So it has energy inherently, and if I let go of it, the energy gets transferred in the form of it falling. And it is the ATP synthase that is able to pick up on this energy transfer and create ATP. And the one way that we can really measure this is by adding a fluorescent tag called rhodamine 123. So how rhodamine 123 works, and this is the basic structure of rhodamine 123, is that the protonated form of rhodamine 123 fluoresces. So if you notice over here, this nitrogen here has been protonated again, giving it a positive charge. And when this happens, it's going to fluoresce. So we can measure this fluorescence using flow cytometry. And this is an example of the flow cytometer that we have here at school. And essentially the way that this works is we suck up yeast cells with this rhodamine 123, this fluorescent tag, one at a time, and we shine a laser at it. And from that we can use different detectors to determine some different things about it. For instance, we can talk about how big or how complex it is. So in this is an example of a scatter plot that you may get from a flow cytometer. Each one of these dots represents an individual yeast cell, and within this picture there are 30,000 of them. So on the y-axis here, we have side scatter, which if we go back, is this guy right here. The laser shines on the yeast, some of it goes through and gets detected by the forward detector, and a lot of it gets scattered off, and then you can detect all of the scattered light or specific wavelengths, depending on what you're looking for. In this case, we're just looking at all the light that's being scattered, and that's a measure of how complex the cells are, how much stuff is in them. On the x-axis, we have forward scatter, and forward scatter is just a measure of how big are these cells. So as you go over to the right, you are getting bigger and bigger cells, and as you go up, they're getting more and more complex. This becomes important because we can select for cells that we specifically want to look at. So for instance, if you have cells that are exceptionally small but very complex, that may not be representative of what we're trying to look at. Okay, so another thing that we can look at is FL1 versus FL3. And it is important to note that both of these are a geometric scale. Just because the variation within the mitochondrial membrane potential, the variation within the amount of fluorescence is so large that the only way that it makes any sense is if we use a geometric scale on it. So on the y-axis, we have a red detector, or FL3, and this basically allows, once we add propydium iodide, which fluoresces red, it allows us to determine if the cells are alive or dead. So if you look, as you go up, these cells are probably pretty dead. On the x-axis, we have a green detector, FL1, which is going to detect rhodamine 123, and this is going to be the way that we measure mitochondrial membrane potential. So the visual representation of FL1 is going to be using a histogram. On the y-axis, we just have the number of cells. On the x, it's just going to be a FL1, which I said is a geometric scale. And the data that will be presented from this point on is by taking the averages of this peak here for whatever test we were running. Okay, so some of the questions that can be answered using this method. Generation 1 beer tastes different than some of the subsequent generations. Well, why is that? Well, in this case, if we are assuming that perhaps there are more flavor compounds being produced later, maybe mitochondrial membrane, we can use mitochondrial membrane potential to figure out where the NADH is being used. Do different worts affect yeast health? So some previous related work, some other students have noticed some generational effects, some differentiation of yeast strains, and the effect of the media in which the yeast was grown on mitochondrial membrane potential. So for instance, the first thing is that the mitochondrial membrane potential differs upon strain. So each beer type has a different yeast. So if you're making an ale, you'll have an ale yeast. If you have a lager, you'll have a lager yeast. There are different strains that are bred for specific flavors that can be correlated with specific styles. And of that, each one has a different mitochondrial membrane potential. So if you look here, you have your ale, which is typically pretty high in flavor compounds, and it has an exceptionally high geometric mean of fluorescence. Now I want to point out that a high geometric mean of fluorescence means low mitochondrial membrane potential. All right? So then you continue going down and you have a cask ale, which is English, a Trappist, that's a Belgian beer, and then a lager, obviously, that is fairly crisp and clean. The second thing to notice is that mitochondrial membrane potential varies over generations. So on the x-axis here we have the generation number. On the y-axis we have the geometric mean of fluorescence. Remember that higher fluorescence, lower mitochondrial membrane potential. And we see that at Gen 5 here, there is a peak in fluorescence which correlates to a lower mitochondrial membrane potential. So for the first 4 generations, mitochondrial membrane potential was relatively stable, and then it shot down and then recovered to where it was before. And then mitochondrial membrane potential in different media. Here we have 3 different strains of the same species of wild yeast. And you'll just notice that, let's take 210 for an example, that grown in wheat wort, it has a geometric mean of fluorescence of 52 versus Scottish of 47 and stout of 36. Now it's important to note that the geometric mean for stout for this 210 was less than either Scottish or wheat, but that isn't necessarily always true as you go down the strains. For instance, if you look at this 212, you'll notice that it has a wheat geomean of 45, a Scottish of 52, and a stout of 47. So the takeaway from this is that the type of media that a yeast is grown in will affect its mitochondrial membrane potential, and that it is not affected all in the same way. It is strain dependent.
Greg, are these all lager yeast? Are those lager yeast?
Yes. So just a little bit of a method overview. We did a couple different types of sampling in some different areas. Here we have a lovely fermenter, so this is the primary vessel in which fermentation occurs. The cone is one area that we were able to sample at the very bottom. This is typically where a lot of the yeast collects. We have the body of the fermenter, which the port for is right here. And then you have the muck, which is essentially everything that is getting blown off the top and into these gigantic buckets. So the first, we ended up doing a 1 to 10 or 1 to 50 dilution, depending on where it is that we took the sample, remembering that the cone is far more concentrated than the body or the mug. And then we subjected it to flow cytometry using 5 micromolar of rhodamine 123 and 15 micromolar of propydium iodide. So the first thing to notice is that there is a difference in mitochondrial membrane potential depending on the level of the fermenter. So on the x-axis here we have sampling location with the cone. Remember that that is here. The fermenter body, which is here, and then the muck, which is everything that's getting blown off. And then on the y-axis we have geometric mean of fluorescence. Remember, higher fluorescence, lower mitochondrial membrane potential. So we notice that the yeast that are contained within the body of the fermenter have a vastly lower mitochondrial membrane potential than either the cone or what's getting blown off the top. This would indicate that the yeast that are in the body of the cone are probably using a lot more of their NADH to create flavor compounds. We also then tried to replicate the mitochondrial membrane potential over generations, and I will point out that this was all collected from the body of the fermenter. So if we go back, it was all collected from here. That will be important later. And these represent technical replicates. So they're not biological duplicates, they are just a measure of the variation that occurs within the machine. So on the x-axis here we have generation, and we have on the y-axis we have geometric mean of fluorescence, and we see that there is a uncharacteristically large amount of fluorescence coming from Gen 4. Now, if you overlay that with the previous data that had been collected, the blue line here is going to be the data that I collected from the body of the fermenter, and the red is going to be the data collected by the previous student from the cone of the fermenter. So they're collected from slightly different places. But we can still kind of observe a similar pattern in that there are a couple of generations where it stays relatively low. There's one that shoots up and then it shoots back down again. The last thing that we notice is that mitochondrial membrane potential varies depending on the style of beer. So you'll notice here that once again we have average geometric mean of fluorescence on the y-axis, and these are biological duplicates. And we have beer style on the x-axis. What's particularly interesting is that the IPA and stout have a higher mitochondrial membrane potential than either the Scottish or the honey wheat. This is particularly interesting because IPA and stout typically tend to be start with higher original gravities. That is, that they contain more sugar at the beginning of the fermentation than either Scottish or honey wheat. Now, if you relate that back to what we talked about earlier with aging, so we're ending up with the honey wheat probably creating a lot more flavor compounds, but also is going to live longer. Remember that less NADH means that it has an increased longevity. So we talked about generational effects, we talked about differentiation of strains, another thing, and then we finally talked about the fact that the media for which the yeast is grown and affects its mitochondrial membrane potential. Well, can we see this on a sensory level? And what ended up happening is we did some sensory analysis, not on the batches that we tested, but on previous batches for vanilla stout, for IPA, and for Scottish. And we incubated a bottle at 30 degrees Celsius, and we refrigerated one sample for 3 weeks at 10 degrees Celsius that came from the same bottling run. And then after 3 weeks, we asked the question, how stable is the flavor? And the way we figured this out is through a triangle test. So essentially you offer an assessor 3 glasses, you fill one with one sample, it doesn't really matter which, and the other 2 with the other sample. So for instance, A in this case could be refrigerated, B could be incubated, and then you randomly place them in front of the assessor and see if they are able to guess correctly which one is different. From this, we were able to notice a little bit of differences within which styles consistently had problems passing this 3-week flavor stability test. The idea was that if the flavor was stable, there would be no difference between the incubated and the refrigerated sample. And what we found is that vanilla stout, by and large, had about a 90% pass rate. IPA had about just over a 60% pass rate, and Scottish had about a 30-35% pass rate. So if you'll remember, the Scottish ale had the lowest mitochondrial membrane potential, and it has the lowest pass rate after 3 weeks for an incubated versus refrigerated triangle test. Okay, so some conclusions. Different styles affect mitochondrial membrane potential. It varies within the level of the fermenter. And some differences were observed in different generations, but it's important to note that we used different yeasts and they were sampled from different places. And some potential, why do brewers care? This can affect brew pitching procedures, depending on whether or not these changes within the mitochondrial membrane potential of different worts carry over to the next beer. So let's say that you have a honey wheat, low mitochondrial membrane potential, and you put it into a stout, which has a high mitochondrial membrane potential. Do you still see a higher mitochondrial membrane potential in the subsequent batch after passing it through something that instigates a high mitochondrial membrane potential? And then it's just a general method of determining yeast health. So this can be used for quality control. If you set a basis of this is what you expect to see and there's something significantly different, you can probably determine that there's something wrong with that fermentation. And then it would also be very powerful if we could link this mitochondrial membrane potential to sensory analysis. So if you had the same style, 2 yeasts with different mitochondrial membrane potentials, and you came up with a different sensory analysis result, that would indicate that this is indeed playing a role in flavor compound production that is tangible at the other end and is something that we should be looking at in the brewery. I would like to thank Dr. David Hall for his help on this project. The wonderful people over at Stone Cellar Brew Pub, specifically Steve and Bobby, thank you Bobby, and Connor, and then Dan Martin down in the stockroom for all his help. So, is there any questions? Yeah.
Okay, so you said that yeast has been cultured for thousands of years for making beer.
Yes.
So when you're coming up with a new beer, I don't know if it would be a brand or a flavor, are you trying to manufacture existing yeast strains or are you trying to find a new one?
So are you talking about if I'm trying to create a new beer, if you're trying to create a new beer style, I'm not exactly sure how that would work. If you're talking about just trying to create a new beer within a style, there are several different yeasts that are associated with that style which have slightly different attributes. So for instance, they maybe flock a little bit earlier or they give different flavor compounds. Basically, the strain of yeast that you use can be used as any other ingredient, such as grains or hops. Does that answer your question? Okay. Yeah, Veronica.
Okay, so why would the membrane potential go really low in like a middle generation and then go back to being high? I don't really understand why this is like blind dip and then it goes back.
That is an excellent question, and you know what? I don't know. We don't know. It's something that we've observed a couple of times, and we're not exactly sure what to make of it. Theoretically, there's some type of maybe selection event, or there's something happening to the epigenetics of the yeast. We just don't know, and this is hopefully something that we'll be able to continue looking at in the coming years.
And you showed it for two different strains as well.
2 different strains and 2 different levels in the fermenter.
So what would you do to chase them?
To chase...
What would be the next experiment?
What would be the next experiment? The next experiment that I would be interested in seeing the results from is one, just following a yeast from the initial pitch and then watching it evolve from generation to generation, basically to see, what I'm most interested in seeing is, does this mitochondrial membrane potential stay the same after you pass it through a wort that sponsors a higher mitochondrial membrane potential? So theoretically, if you had a let's say a honey wheat that has a low mitochondrial membrane potential, and you wanted to recover that yeast, give it a little bit higher mitochondrial membrane potential, you could throw it through a stout, and that would if that fixes the problem, then we don't have to worry about hitting specs. Does that make any sense? So you would basically establish what the average mitochondrial membrane potential is for every style, and then you would figure out ways that you can manipulate it in order to maintain that consistency, because it's really the consistency of flavor that we're concerned about. If it's a matter of they're producing lots of flavor compounds, that's great. We just want it to be consistent.
So basically like crop rotation.
Yeah, essentially. That would be the idea. The other thing is that we have also been looking at some mitochondrial genomes to take a look at whether or not we can find genetic markers for specific yeast attributes and specific flavor compounds. So basically looking through and figuring out if, hey, this gene correlates to finding phenolics in the beer. I don't know if you had a chance, I think it was CNE that had an article dealing with yeast genomes in the brewery, which is very cool if you have not had a chance to check it out. Anything else? Yeah, what's up, Winter?
Can you just define generation for me? So I'm not sure if you mean
Yes.
the budding scars or between batches.
Between batches. So when we refer to generation, generation 1 is when we originally take a yeast, brand new yeast, and we put it into a beer. After that beer is done, when we take the yeast back out and put it into a new beer, that's generation 2. So basically, we recycle yeast for anywhere from 6 to 9 generations, depending on how it's doing. Any other questions? Yeah.
So when you talked about flow cytometry,
Yes.
You talked a little bit about the size of the yeast, and
Correct.
like, what would that do for you? Why would you want to do that?
Let me pull that back up momentarily. Okay, so this is the slide that you're referring to, right? This is maybe not an example of some of the things that we saw. We saw a lot of variation